So I'll say welcome everyone to the November National Newborn Screening Pilot Monthly Webinar. My name is Dr. Amy Brower. I'm a medical geneticist at the American College of Medical Genetics and Genomics, and I am the PI for the Newborn Screening Translational Research Network. And so this month, I'm really excited about our agenda. But before we get into that, just a few housekeeping items. Please mute yourself while others are speaking and use the chat window to ask questions during the presentation. We'll open it up so you can have some active dialogue after the presentation, unless Dr. Bedoyan, our invited speaker, tells us differently. The call will be recorded and posted on the Newborn Screening Translational Research Network YouTube site, and the link is there, or you can visit our website and search for the YouTube link. If you have any issues or have any questions accessing any of that, please feel free to reach out to us, mbstrn@mbstrn.org. So with that, I would love to welcome everyone again to the November call and see if there's anybody new across the teams that would like to introduce themselves. Okay, we've got a lot of old timers here, which is great, including myself. So today we're really excited to welcome an invited speaker. Dr. Javier Bedoyan is an associate professor of pediatrics in the Division of Genetic and Genomic Medicine at the UPMC Children's Hospital of Pittsburgh and the Department of Pediatrics at the University of Pittsburgh School of Medicine. He's a clinical and biochemical geneticist investigating the genetics and pathophysiology of several inborn errors of metabolism, including disorders of pyruvate metabolism and a focus on mitochondrial pyruvate dehydrogenase complex deficiency, known as PDCD. Dr. Bedoyan has achieved local, national, and international recognition for excellence in patient care and for his novel scholarly work to improve patient care and health outcomes for several complex inherited disorders of energy metabolism, including PDCD. So join me in welcoming Dr. Bedoyan.
Thank you, Amy. That was a generous introduction. I don't see the slide. Shall I? Oh, shall I? Is the slide from your end? Oh,
Yep, I can do that. And then can he manage the slides, Jennifer, while I share, or will Dr. Bedoyan have to share?
I think he'll have to share his own slide unless he wants to just direct you to the next slide.
oh,
If he shares slides,
yes, I can do it. Just 1 second then.
Thank you. And I'll just add, as Dr. Bedoyan's finding his slides, after his presentation, we'll have a good bit of time for question and answer sessions, and then we'll get an update from the folks on the call today who are running active or completed pilots, or just really open it up to any discussion of newborn screening pilots that are all on your mind today. And then the MBSTRN has a few updates that we'll go through at the end. Our partners, APHL NewSteps, didn't have any updates for us this month after coming off their really exciting annual meeting last week.
Okay, so shall I share my
Yes, go ahead.
screen? Okay, I'm gonna share my screen. Whoops, I'm gonna share this one. Share. Can you all see the slide?
Looks perfect. Thank you.
Okay, let me expand this and let me remove subtitles. Okay. And then let me minimize this one, and then I'm ready to go. Okay, it's a pleasure being back in this forum here, and thank you, Amy, for inviting me back to talk a little bit more about pyruvate dehydrogenase complex deficiency. I gave a talk at this forum about a month and a half ago and introduced this complex and this deficiency, but I'll review a little bit again for those who were not in attendance at that time, and then bring you up to speed with what were the lessons we learned from our pilot newborn screen study that we put together in Ohio when I was there in Cleveland, and we published on that, and how we're expanding the utility of the screening biomarkers that we found and used for that pilot study, and how we're expanding that in the state of Pennsylvania. I have no conflicts of interest to report. And so, like I said, the outline is, what is PDC deficiency? It's biochemistry, enzymology, its clinical features, the molecular genetics, and E1 structure. E1 is one of the components of the multi-complex pyruvate dehydrogenase complex. And I just would like to sort of stress and have you think not in sort of two-dimensional way, but in more three-dimensional, non-static or motions of how this three-dimension structure, the E1 complex, works, and how missense mutations, disease-causing missense mutation, is altering the structure, so that we can have an appreciation of how to tackle target-based therapies for this disorder, which has not been tried before, that we're doing in our laboratory here. And so then we will talk about the therapeutic options that are out there right now and the things that are up and coming. And that will be a good segue to sort of go back into pilot newborn screen protocol and the approach we use and its limitation, because it was hard to put that together in Ohio, and I think it's going to be harder to put that together in any other state, and the utility of the biomarkers that we used, and how we're expanding those in a cohort of patients from UPMC spanning 11 years. So we've identified about 11,000 patients who've had amino acid analysis done and who've had a confirmed molecular and enzymatic diagnosis in a large fraction of those, and how we're approaching doing the same thing at this level here, given the limitations we have in changing the type of metabolites we analyze in dried blood spots from a newborn screen lab in the state of Pennsylvania, and for that matter, other states as well. Okay, PDCD is a mitochondrial neurometabolic disorder of carbohydrate oxidation that mostly affects the brain and leads to a decreased ATP production and energy deficit. So a one-liner would be PDC is a mitochondrial neurometabolic disorder of energy deficit, and that's key. It's a major cause of primary congenital lactic acidosis. It's the second most common genetically resolved mitochondrial disorder in the North American Mitochondrial Disorder Consortium Registry, second only to the Golgi-related mitochondrial disorders. And from our pilot study, we know that at least 1 in 40,000 live births annually will be affected with PDCD in North America. And that roughly translates into about 90 newborns annually in the United States having PDCD. It's this incidence is about similar to what we're seeing in GA1 and very long chain fatty acid oxidation disorder, VLCAD. PDC is a complex that sort of sticks, falls between the end of glycolysis and the beginning of the TCA cycle. And so the end of glycolysis is a 3-carbon keto acid, pyruvic acid, and the product of PDC, which is an irreversible reaction, is acetyl-CoA, a 2-carbon CoA, that feeds the TCA cycle at 12 o'clock and then allows for generation of NADH and FADH as reducing potentials that then feed into the electron transfer chain and generate ATP that way. The one other the dehydrogenase, the pyruvate dehydrogenase, also shares many similarities to the glutarate dehydrogenase complex, the alpha-ketoglutarate dehydrogenase complex, which is about 6 o'clock here in this figure. And keep that in mind, because that's something we'll come back to show you how we can take advantage of that dehydrogenase to understand other types of pyruvate dehydrogenase complex deficiencies. Okay. I'm trying to block the thing here. Okay, that's fine. Can I go back? Yep. So when the complex is deficient, what happens enzymatically and clinically is pyruvate accumulates. And when pyruvate accumulates, alanine accumulates because it's a transamination product of pyruvate. And then when pyruvate also accumulates, then lactate dehydrogenase generates lactate. And so the kid will have lactic acidosis and hyperalaninemia. High lactate over 3 millimolars usually causes inhibition of the primary enzyme involved in proline metabolism, which is proline oxidase. And so proline starts building up. The important thing to note, unlike other primary mitochondrial conditions, primary meaning more having to do with the electron transport system and the OXPHOS, is that the lactate to pyruvate ratio is usually normal. It is not usually, it is normal. Some folks call that range 10 to 20. I've seen patients between 20 and 25 that are bona fide pyruvate dehydrogenase complex deficient patients. So my upper normal is 25, but anything above that is much more in the realm of primary mitochondrial disorders and electron transferase defects and such. The clinical picture is quite broad. It has neonatal onset, infantile form, and late onset in children and adults with milder presentation sometimes. And again, the clinical features are variable too, but there's some common parts to it. If you're seeing in a typical presentation of PDCD, which would be hypotonia, 89% of them, developmental delay, 83% of them, ventriculomegaly, 85% of them, corpus callosum abnormalities, up to half of them, seizures, again, up to half of them, Lee syndrome, about a third of them, and ataxia, about a quarter or so. And the median age of diagnosis is about 12 months. So that's quite delayed, and we need to do something about that. Females have a better survival than, but surviving females are more severely affected than males. And at least from the study from back in 2012, 90% of them would die by 4 years of age. And here's 3 clinical scenarios of how they may present. This is a female 13-month-old with generalized hypotonia, developmental delay, paucity of white matter, a thin corpus callosum, patchy enhancement of the caudate and lentiform nuclei bilaterally, and an elevated blood lactate and pyruvate with a normal ratio. Another female is a day of life 28 x 33-week preterm with dysgenesis of the corpus callosum and elevated blood lactate. And then finally a male who's full term newborn with generalized hypotonia and a brain MRI showing thinning and hyperintensities of white matter, and also elevated blood lactate and pyruvate and a normal ratio. Of course, this is a PDCD presentation, so I would suggest that we do that all of them will have a functional PDC deficiency, and indeed, when you measure them in lymphocytes or fibroblasts, you would at least have one of those cell types to be low, and we see that low in number 1, low in number 2 in terms of fibroblasts, and low in number 3 in terms of lymphocytes. We know about tissue and cell type variability in this assay, and we know about gender-specific variability in these assays. So this is not surprising. We've done a second assay, which is the alpha-ketoglutarate dehydrogenase complex assay in both 2 and 3. 2 is low and 3 is normal. So the fact that 2 is low means that this, at least genetic basis for this functional deficiency of PDC, is different than the male, and different than likely the first one, although this was not done. E3 deficiency is excluded by E3 being normal, and E3 is one of the components of the PDC complex. All of these kids were started on a ketogenic diet because of the functional deficiency in PDC and the presentation. One of them lived, the other two died very soon after the start of a ketogenic diet. They had 3 different genetic bases for the functional deficiency. One is the PDH-A1, which is seen in up to 88% of all PDC patients. Another is LIAS, lipomide synthase deficiency, and we'll see why. And then one is another secondary type of PDC deficiency, the enol-CoA hydratase deficiency. And we've subclassified PDCDs into either primary specific, depending if the genes are from the main primary common genes of the PDC complex, primary generalized, that have to do with gene products that are involved in modifying certain parts of the complex, or secondary, where other mutations in other parts of the metabolism pathways like TCA or valine metabolism or fat metabolism indirectly affects the complex, either at the active site or the modifying, and this causes dysfunctions. So this is the complex, and so this is the E1, and E1 is a dimer of heterodimers composed of PDH-A1 and PDH-B gene products, E2 from DLAT, and then E3 is the dihydrolipomide dehydrogenase. The thymine pyrophosphate is a cofactor and an active cofactor. It needs to bind and be activated for the first step of the reaction, which is a decarboxylation reaction of pyruvate to release the CO2. And then the acyl group then is transferred to the E2 component through this lipoyl group, which is its 8-carbon moiety, which one of the LIAS deficiencies interferes with the synthesis of this lipoyl group. And then eventually an acyl transferase and to the CoA as acetyl-CoA product. And then the lipoyl group is reoxidized by the E3 component, and an NADH in the process is generated. There are 2 regulatory components to this. So a kinase phosphorylates certain serine groups within the active site of the E1 component and inactivates it, and phosphatase dephosphorylates it and reactivates it. So if one is thinking of ways to approach therapeutics, if the PDC complex is deficient, let's say most commonly from a PDH-A1 mutation, then you're really not generating the product. So replenishing the product is a key component of an approach for therapeutics. If you have one of those regulators being defective, then one approach would be either having a product, for example, if the phosphatase is defective, so you're not able to remove the phosphate, a small molecule that inhibits the kinase is an approach where you can maximize the activity potential that the complex would have because of a defect in PDH in the phosphatase. Another approach would be that one we are using or approaching is to use a target-based approach with small molecules to restore the activity of the E1 complex, so that pyruvate now has a better way, better potential to generate the product in such a way that you replenish the potential for and restore the energy deficit that the individual has. So different components are shared by different dehydrogenases, like the lipoyl group, which is the red arrows, are also shared by the alpha-ketoglutarate dehydrogenase, the oxoadipic acid dehydrogenase, which is the proximal dehydrogenase right proximal to GA1 enzyme, the branched-chain ketoacid dehydrogenase that's responsible for MSUD, and the glycine cleavage system that is responsible for glycine metabolism. Similarly, the the E3 component is also part of the component of these dehydrogenases and also the complex, the glycine cleavage complex. So when you have mutations in DLD, it's not only limited to how pyruvate is metabolism to make acetyl-CoA, but it interferes with other metabolic components of branched chain amino acid, glycine cleavage, etc., that would be much more generalized than the specific nature of any component that is part of the main complex. And so this is how we've classified them into at least 3 subclasses. And we spoke about PDH-A1. We spoke about the LIAS being in a more primary generalized subclass, and then the short chain enol-CoA is much more secondary class of PDC deficiencies, along with the SUCLAs, the succinyl-CoA lyase deficiencies, which is about 7 o'clock of the TCA cycle right next to the alpha-ketoglutarate dehydrogenase. Tafazin gene, the TAZ, which makes tafazin, which is an acyltransferase that helps remodel cardiolipin, which is component of the inner mitochondrial matrix, is also an important component that the recent evidence shows that it helps tether the complex to the inner mitochondrial membrane within the matrix. And deficiencies of TAZ also cause functional PDC deficiency. This is the E1 component, which is a dimer of heterodimers, and these are the 2 active sites, loop A in the alpha-1 subunit, and the other loop, the symmetric loop, in the alpha subunit right here, and with the thymine pyrophosphate shown as spheres here. And components of this loop A and loop B are important regions of the binding of the TPP and also sites of serine where the kinase phosphorylates and inactivates the complex. So it's a multi-enzyme mitochondrial matrix complex. More than 38 genes are known to be associated with functional PDC deficiency, but up to 88% of cases are due to this X-linked PDH-A1 gene. But 5% of the remainders are due to the other primary specific genes, PDHB, DLAD, PDP1, the PDHX. And of the cases with PDH1 mutations, about 60% are due, 55 to 60% are due to missense mutations. And the arginine residue replacements constitute about 40 to 50% of all these missense mutations. And the recurrent replacements of 3 arginines, specific arginines, R234, 273, 349, constitute about up to 80% of all these arginine replacement. And there's gender predominance. The 273 site is very close to the thymine binding site, and it's very much female predominant. The R349 is much more of a male predominant, and we've shown that there is an allosteric impact of these replacements, and we'll briefly talk about those. And the other two last sentences, I'll be mentioning those in the next slide. So this is 3 different ways of ascertaining the databases for PDH-A1 mutations, one through the CITEM database in Cleveland, one through ClinVar in Genome AD, and one through a recent cohort of DCA of PDC patients on DCA trial. And altogether, we have something like 250, 260 subjects with missense mutations, and you can see between 40 and 50% of them will have an arginine change, and that the arginine 349 constitutes about a quarter of all the arginines that are replaced. So if you have a pretty big center that at least has 10 to 15 patients in your center with PDC deficiency, I would predict at least one or two of them would have an arginine 349 substitution. And we'll see about the last two slides with the next slides, two rows with the next slides. So Nicole Dusich is a medical student who's worked with me for 4 years, and she was interested in seeing how the missense mutations and nonsense mutations distribute themselves among the different exons, the 11 exons of PDH-A1. And this is just looking at a two-dimensional way of where these mutation falls, and indeed most of them fall in this between exon 5 through 7, the missense ones. She was also more interested in looking at how these mutations are, where they are located in the 3D structure, and how they're affecting the 3D structure. So we started looking at what is called solvent accessibility surface area. It's the measure of how accessible an amino acid residues is in a structure to the solvent. And one can measure that particular amino acid in the E1 structure as a complex, or in the subunit alone, like the alpha subunit alone. And that is an important way of comparing both, because then one can not only see how much of solvent inaccessible one residue is, but how likely that residue is involved in subunit interfaces. So the first question is, lo and behold, we saw that most of disease-causing variants are buried, and only a small fraction of them were accessible to the solvent, as opposed to neutral variants from the databases. So 86%, 85%, 86% of all the variants are solvent inaccessible, and that's also true for PDHB, although we don't have as much mutations out there in the databases as there is for PDH-A1. Then, like I said, if we compare the solvent accessibility in the E1 complex as dimer of heterodimers and each of the subunits alone, then we can get an idea of how many of those buried amino acids are involved in subunit interface contacts, and indeed 30% of them were involved. And those were of interest to us because we were interested to know how those amino acids that are involved in keeping the subunits orientations intact and linking them all together in terms of communication, how they're breaking down when you have a missense mutation. So to convince ourselves that indeed those were involved in subunit interface contacts, what Nikki did is also walked along the amino acid. This red one is the PDH-A1 component, so the alpha subunit, and then the blue one is the PDHB or the beta subunit. So she went along each of the amino acids and asked herself, what would be in the complex? What would be the nearest neighbor to that amino acid less than 5 angstrom away? And then as she's walking through, anything that's blue horizontal is the nearest neighbor. So in this case, it's the beta 1 subunit. In this case here, it's the beta subunit. And the red horizontal is the alpha 1 subunit. So there are regions of amino acids that are nearest to the alpha unit within the beta subunit, and so on. And there are regions of the alpha 1 that are in close proximity to those amino acids on the alpha subunit. And lo and behold, all of those disease-causing replacements that were part of the subunit interface contacts that was shown in the previous slide, this 26 group, fell within those regions that were nearest neighbor in this analysis. And also the same where for PDHB mutations, although we didn't have as many PDHB mutations to analyze as we do for PDH-A1. These are the regions that are nearest neighbor. And we asked our question, and we know the clinical phenotype of all these individuals because of these are all reported. And we asked the question, if we have two nearest neighbor amino acids, one in one subunit and one in another nearest neighbor subunit, if we have a mutation in one and it presents a clinical phenotype, is it likely that the corresponding neighboring replacement also causes another similar phenotype, because PDC is so variable? And indeed, we were lucky enough to find two pairs that from which we've had clinical information complete. This tyrosine 132 in the alpha subunit and the methionine 71 in the beta, and also two amino acids in alpha and alpha 1 were nearest neighbor. And I'm going to be talking about one common amino acid replacement, which is the R349, which constitutes about 10% of all the amino acid missense mutations in PDH1. And its nearest neighbor is E-glutamate 281. We don't have a patient with that, but you'll see that the molecular dynamic phenotype that is presented by mutations in this E21 resemble that of 349. So this is the first pair where these patients both would have neonatal lactic acidosis and lead disease and seizure. And the other pair is where they both have neonatal lactic acidosis, lead disease, and severe psychomotor retardation as specified in their clinical. And they're published on different the years and different labs. And so what we had a strong interest in this R349 for two reasons. And we're comparing with another amino acid, tyrosine 185, that's involved in this the SSIC, the subunit interface contact. And the reason we chose those two, this one is pretty far away from the loop A active sites, and this is a little bit closer. But all of these pink ones are involved in SSIC. You can see the distribution is very much different than a random distribution. And so Hattie Gökhan, a postdoc, was interested in looking at the dynamic implication of changes in that arginine 349 and tryptophan 185. And here's the molecular essential motions of the wild type complex. You can see that this loop A, green loop here, is highly movable, and the same way with the loop A behind this pink line here, and that things are moving. These are under physiological, as much as possible, physiological conditions that the matrix would find itself. And so Hattie then did a relative fluctuation, looked at the fluctuation of the backbone alpha carbons, and looked at the motility and compared that relative to the wild type. And so anything that has peaks here that are close to 2 angstrom or more implies those are significant mobility changes upon replacement with either the common histidine or the cysteine that we find in patients, and in both the subunits, so alpha and alpha prime. So this one generates a genotype that would be found in males, for example, where because males only have one X PDHA1 gene, then both the alpha and alpha subunits would have the same replacements. And we've also done the same things with only one replacement to mimic what you would see in much more of a female gender. And so then we asked ourselves, what is the nearest neighbor of the R349, which is the E281? And we don't have viable humans with those mutations, but we predicted the delta G free energy that anything that's value of negative, at least -1 or what at 1.3 implies significant destabilization, and anything positive is stabilizing. And we know that these two are destabilizing and they result in low PDC activity in vitro. And we had no idea of the other ones. But then Hattie did molecular dynamic fluctuation analysis using these replacements of E281 to arginine and E281 to cysteine. And indeed, again, the only major difference is in these loop A regions. So these are the nearest neighbor to the R349. and we're seeing the same sort of a dynamic change that we were seeing with R349. The implication being that if we do find a patient with the E281 replacements, they would clinically very much probably look like what patients with the R349H or R349C would have. And then we went into one other way of analyzing this communication network between different subunits. It's called eigenvectoral centrality analysis, looking at what these mutations are doing in different parts of the various subunits and which amino acids are being implicated in that. And indeed, and comparing that with the wild type, of course. And then indeed, the regions that we were seeing communication networking breakdown are the same regions that are involved in subunit interface contacts by SASA analysis or nearest neighbor analysis. And those are the regions that have the most of those mutations that are involved in subunit interface contacts. So we're trying to model, come to a picture of how this three-dimensional E1 structure is being disrupted when you have a mutation that are several angstroms, up to 20 angstrom away from the loop A structure, and how these interactions are being compromised. And so, of course, we've identified the R349, and any replacement with either histidine or cysteine forms a pocket, and that pocket is well defined. And so our lab is working to ways to dock a small molecule in that pocket, and then see if that small molecule, when docked and bound, restores the broken interactions between the various subunits and helps restore the activity of the PDC in vitro and also by simulations. So we've seen through this what this complex is. And so the therapeutic approach I hinted to is either providing the missing product, restoring the defective complex component for, let's say, E1 complex, replacing the defective component of the complex altogether by either enzyme replacement or gene therapy. And gene therapy is something that's being looked at now, at least two different labs, one in the United States and one in Europe. Enzyme replacement therapy is going to be difficult because of the blood-brain barrier. Or another approach would be manipulating these regulators of the PDC with small molecules that inhibit the kinase, like say DCA, or restore or replace PDH phosphatases to sort of restore the PDC activity appropriately, or provide cofactors of the PDC. This is in case that there's a thymine responsive cases, which are very few out there. So ketogenic diet is a diet that replaces the acetyl-CoA by forming ketone bodies, which pass through the blood-brain barrier, and then are in the brain cells, then are generated. They get converted back to acetyl-CoA, which then feeds at 12 o'clock of the TCA cycle to generate energy. And it's been shown to have positive effect in terms of epilepsy, ataxia, sleep disturbance, speech and language development, social functioning, and frequency of hospitalizations and survival. But its efficacy depends on the birth severity, depending on whether how much of a brain anomalies are found at birth in this child. And early intervention with ketogenic diet, especially those milder cases where they come in at 12 months or 15 months, or more recently, I've seen 18 months and were diagnosed and started on the ketogenic diet at 18 months with, milder phenotype instead of having it done earlier. And the efficiency depends on how ketosis is attained and maintained. Dichloroacetate as a small molecule, and it inhibits the kinase, and in so doing maximize the activity. And the phase 3 trial has enrolled at least 32 subjects now. It's not recruiting anymore, and data is being analyzed. thiamine responsiveness is very few. Other options, phenylbutyrate, glycerol phenylbutyrate, the trial has ended and there's a mixed effect as far as I can hear from others. Triheptanoin, which is an anapyretic agent, and anaplerosis is an agent where they restore these intermediates of the TCA cycle. And we hypothesized that that would be a nice agent to use for primary specific PDC D, and we wrote a grant and we got funded by an industry, and we will start the trial early, either late first quarter of next year or early second quarter of next year. And target-based small molecule options are investigated in our lab. And then again, like I said, gene therapy not only in Europe on a patient, but then experimentally on mice in the United States being pushed over. So pilot newborn screening and their approaches of that. So why is it so important? This is data from 13 patients here in the UPMC system, where we looked at when the first serum amino acid was ever measured in this patient, when the molecular diagnosis was confirmed, and when the ketogenic diet was started. And the median was 10 months for first amino acid, about 17 months for a molecular diagnosis, and about 13 months for starting a ketogenic diet. So this is very openly showing you how important it is to be able to pick up these kids early, so that at least the conventional untargeted therapy, which is ketogenic diet, is started on these kids as soon as possible. So the premise being that early diagnosis of this disorder and early intervention with ketogenic diet or other novel therapies would lead to improved development and cognitive outcome, quality of life in terms of seizures, hospitalization, and longer survival. So we knew that alanine and proline are elevated in this condition. We wanted a way to, and both alanine and alanine is not something that's measured in the dry blood spots, and we knew that leucine and lysine are the strictly ketogenic amino acids, which means that they bypass the PDC complex and enters the TCA cycle at the acetyl-CoA level. And so we figured that that these two amino acids, leucine and lysine, would be a new, a good denominators to normalize alanine and proline values to avoid noise from elevated alanine and proline that would be not as obvious. Yeah, and we've looked at that, and alanine by itself, and alanine to leucine or alanine to lysine, and the ratios were much more sensitive for PDC deficient individuals. And this we've done in 22 subjects, 20 of them with PDH-A1 mutations and 2 of them with compound. There were compound heterozygous and PDH-B mutations, and this was plasma amino acid. And we realized that this ratio used in combination was sensitive in these individuals. And we said, let's put together an approach to pick up these kids if we can at the newborn screen level early on. And of course, alanine and proline data are not found in the metabolites that all newborn screen labs would report. And so we worked with the Ohio State Columbus Newborn Screen Lab, and it took a year to work with them. And most of that year was spent more of an administrative and legal and regulatory capacity, instead of actually changing the algorithm of how we detect and measure the compounds. And finally, we were able to identify and quantitate them starting May 4th, 2018. So those are now two metabolites that are added to the supplemental information released from the newborn screen lab. They're not used as part of the PDC newborn screening but the clinicians have this information so that if they want to look at it and they want to do the ratios or look at to see if there's a concern of PDC deficiency or any other mitochondrial deficiency, they can make use of that. And then we obtained de-identified dried blood spot data. And this was also a little bit of a work to get them to release de-identified information in terms of gender, gestational age, birth weight, NICU stay, whether they were on TPN or received packed red blood cell or not, the age and time of dried blood specimen collection, and the alanine, proline, and leucine concentrations. And then we send around flyers to different medical centers that have geneticists and neurologists around the state, so that if they advertising the study, and if they were to have a subject born after May 4th, 2018, that was diagnosed with PDC deficiency or other mitochondrial condition, including organic acidemias and fatty acid oxidation disorders, to send to us to enroll in the study, so that we can now access the newborn screen information and assign a name to that dot of these de-identified samples, because then we would know what the clinical picture is and where that dot will fall in this whole picture. There was no clinic visits, no new laboratory blood draw or clinical tests, because all of these tests were already completed by the primary physician. We were only going to review existing medical records and access the newborn screen test result to identify, put a name to that dot. And so we've done this for over a year period, and we identified 123,414 newborns with non-duplicate and data complete dry blood specimens. And we looked at the greater than 99.9 percentile of either the alanine to leucine ratio or the proline to leucine ratio. And this is the plot. And of those 123,414 dots, only 217 fell in the 99.9 percentile of either one of them. Now, if we, on top of that, add the criteria that we set forth based on the plasma amino acids of patients, and this was an initial attempt by us to set cutoffs and see where we would, where these patients would fall, and then move those cutoffs later on. And our cutoff was alanine to leucine of greater than 4 and proline to leucine greater than 3. So 4 greater than 4 and greater than 3. And means this box here, or would be including this box here and this box here. So we're just limiting it to an and. And indeed, we were able to identify 3 cases of PDH-A1 and 2 cases of mitochondrial condition. And this is the details of those. The 2 mitochondrial children passed away. The 2 PDH-A1 patients were started with a ketogenic diet less than a month of age. And one of them died at 3 months because it was picked up late, and the parents elected to redirect care and not start on a ketogenic diet, and the kid passed away. Afterwards, after this 1-year study, we pushed another half a year and to see what else we could find, and we indeed found tafazin gene, a Barth syndrome patient falling in this group, and a patient with both a nuclear and a mitochondrial mutation that had what looked like a mitochondrial presentation with lactic acidosis and a sepsis kit. So in this situation, we at least said that the positive predictive value of this criteria is at least 3%. And that is much better than the Krabbe disease positive predictive value in Ohio, which is 1%, and very close to the MPS 1, which is also 3%, but not as high as the Pompe disease. And we realized that it's sensitive, but it's not specific for PDCD, because we're also picking up other cases of mitochondrial condition and likely other cases of organic acidemias or fatty acid oxidation. And because we were limited on how the physicians direct these patients to us for us to interrogate and find who they are, we were limited in identifying all the remaining 143 or 140 green spots. So that was the limitation of the study. But we can improve on specificity by adding a second tier molecular test that only specifies for those genes that are components of the primary components of the complex, and not deal with, I'm sorry, other mitochondrial conditions that for which we don't have therapies for. So we're picking up for where I left this study in Ohio. And so when we came into Pittsburgh, then we got approval to look at 11,000 patients over the past 11 years that have had amino acid analyzed. And we started looking at prioritizing the first amino acid prior to any therapy, prior to TPN, any ketogenic diet, any packed red blood cell. And then we prioritize at least patients that have a confirmed MELAS diagnosis, PDC diagnosis, MRF diagnosis, propionic acidemia, methylmalonic acidemia, Lee disease because of the electron transport chain, complex 1, complex 3, complex 6, 5 deficiencies, fatty acid oxidations, and non-IEMN, pancreatitis, encephalopathy, seizure, things like that. And we looked at these alanine to leucine ratio, and we can see that for PDC, this ratio discriminates very well against the other conditions that we have here. Alanine to lysine also does a pretty good job discriminating against the other conditions. Proline to leucine, we didn't have the full data from the prior study in Ohio, but we realized that it does discriminate against, let's say, the MELAS and the MRF and the PA, but not MMA and not the fatty acid oxidation disorders. So this may not be as useful a ratio as we thought it was. Again, proline to lysine, it has some discriminatory potential against some of them, but not as much against some PAs and MMAs and fatty acids. And citrulline, which can be low in primary mitochondrial conditions. And we said, let's look at citrulline to leucine and citrulline to lysine. Again, this has no utility for PDC deficiency. So we have analyzed so far about 167, 200 cases, and this is where their dots fall in terms of proline to leucine and alanine to leucine. So each one of them, we know what the diagnosis is. This is, again, serum amino acid. This is not dry blood spot, but we can eventually translate this into dry blood spot data form. And we recognize there are 14 patients in the UPMC system. All of those are the red dots. And so initially, this is where we would say proline to leucine greater than 3 or alanine to leucine greater than 4 is this gray region of all patients. You can see that we're picking up, if we have this criteria, we're picking up 13 out of the 14 PDC patients. One is false negative.
Dr. Bedoyan, I know Dr. Friedenberg from Texas has a question.
Okay.
Yes, sir. Hi, I actually have two questions.
Yes, go ahead.
One is two things. One is that in siblings that have been identified as a neonate, because an older sibling was affected and put on very early treatment, do we have clinical outcomes for those kids? Like an older sibling was diagnosed with the condition and then the neonate was tested immediately and was put on treatment. Do we have any clinical outcomes?
No, I don't have any data of that. That's a very good question. We have data on Pompe kids with infantile Pompe where one was missed and then the SIB was early diagnosed and quickly started therapy. We have that data on urea cycle disorders with OTC. I'm not aware of a data with PDHA1. I would be very delighted to know about.
And then my other question is, I may have missed this. So in your study design, you looked at de-identified newborn screen spots, but then you got permission from the clinicians to identify them. I was a little confused about how that worked.
No, we don't have permission from the physicians. We have the patients enroll in the study, and the patients will then provide us the permission then to go back and collect their newborn screen information in their medical records.
Okay.
And
So
so
they're clinically identified.
they're
and
clinically
once they're
completely
clinically
identified.
identified, then they enroll in the study.
That's right.
Okay.
So the
Alright.
quicker they're
I think I missed
identified
that.
early on, the
Yeah.
quicker. So you can see the limitations of this study off the bat there. Thank you for those questions. So then the proline to leucine ratio and the alanine to leucine ratio would generate more false negatives, as you can see if we are limiting it to that rectangle there. And so this is the old-fashioned, true positive, false negative, false positive, true negative ratio, picture. And we can see that with this cutoff criteria, we had 13 true positives, 1 false negative, and a sensitivity of 93 and a 52% specificity for this population of 167 cases, with 87 of them in this positive, being positive in this big region here. So then we did the same for different ratios, and we also looked for alanine to lysine to alanine to leucine ratio. And we realized that the alanine to lysine greater than 3 and alanine to leucine greater than 5 has as high and sensitivity and better specificity than using proline to alanine and proline to leucine and alanine to leucine ratio. So we're working on that. That's where we're at right now, in sort of expanding this from only the 1.5–2% of the cases that we've done to 10% or 50% of the cases that we have. And I suspect that the sensitivity will remain high, but possibly the specificity will go down and positive predictive value will go down as we generate more false positives as we analyze those medical records of these queried patients in the future. So a lot of people to thank. The person that I owe this most of my last 12 years of interest in PDC is Doug Kerr. He is retired, and actually I went to his birthday 86th birthday party last Saturday, and that was a real joy to see him in Cleveland again. But then the people I owe this mostly to are the fellows or the techs that work. April Lehman is the fellow that's working on the amino acids at UPMC. Robert RJ is the tech that works on the in vitro assay of using the small molecules and restoring the activity. Nikki Dusage is a 4th-year medical student at Case, and Anisha is a 2nd-year medical student at West Virginia. And then my colleagues in the Department of Chemistry at Carnegie Mellon, Alexander Isayev, her postdocs that we share, his postdoc that we share, and then Polina, his graduate student that is doing a lot of the docking and binding work, and the pharmacology colleague of mine back in Jason Mears, who started us with this molecular modeling. And I shouldn't forget Rosemary Hodge. She was the one who really worked with me very hard and convinced or made the committee agree to moving ahead and releasing the alanine and proline data from dried blood spot in Ohio State. And now Ohio has those 2 metabolites as a supplemental data. And she's now at the CDC in one of the directors of their lab over there. And this is something that April very kindly put together, because I said, because they were frustrated in how slow things are moving. I said, I just need more minions. And then she couldn't stop from drawing this. And then I am of course, grew, and then they are the minions, and I need more of them. But they've been superb now. And that's all.
Thanks so much, Dr. Bedoyan. And I think one thing that your talk reminds us of the really important role that newborn screening programs play in advancing research.
Yeah.
So even though you study clinically identified cases, it was really the newborn screening lab and the partnership with Ohio that led many of these efforts to go forward. So with that, I know we're at the top of the hour, but we will continue for a little bit and see if anybody wants to unmute themselves and ask any questions. I know you'll always be available in MBS TRN, and now that we've made this connection to all the states, we really want to make sure that we keep you involved and helping us move things along. So with that, I'll see if anybody has any questions. So I'm going to go back to your Minions poster or picture and just go forward. And I think, one thing we're trying to do is make sure that we highlight experiences like you had, Dr. Bedoyan, with working with the Ohio program. And maybe we'll have some follow-up discussions with you on, what made that happen. So it was sort of a happy, collaboration and a really fruitful collaboration between your group and the state program. So with that, I'll just go through a few updates from the Newborn Screening Translational Research Network. So I'll turn it over to my colleague, Dr. Ki Chan. So Ki, take it on, and I'll advance your slides for you.
So hello, everyone. So thank you, Dr. Bedoyan, for that wonderful presentation. And I can imagine that we'll continue this discussion afterwards. And also visit our MBSN forum, where Dr. Bedoyan had also shared his slides from the Newborn Screening Research Summit. And that's another way to connect with him offline as well. So I'm happy to share that MBS Chen had led a special issue in the American Journal of Medical Genetics, the seminar in medical genetics. This was the June 2022 issue. Some of the articles were published as long away, and so now all the articles are published most recently. So you can see that this is our table of contents that has a mix of commentary, research from pilot studies, research from on next generation sequencing, and also we have articles written by advocacy group. So this is an issue that's specifically focused on newborn screening research. Like the last type of special issue was maybe published like a decade ago. So this is a very special edition that we hope that you subscribe to it, download some of the articles, visit this website. So this is a special issue that Dr. Bauer and I were the guest editor for this issue. And if you look through the author list, you notice that many of these authors were from our previous network and Newborn Screening Summit. So this is one of the goals, besides presenting your work at our summits and meetings, is that collectively these authors and these researchers submitted to the special issue. So we're hoping to maybe have another issue that's going to be similar like this to showcase your impact in newborn screening research. Dr. Brown, the next slide. We also want to highlight that we have our newest episode and our newborn screening spotlight with Dr. Zenze, also known as Mike Hugh, who is the co-founder and president of Project Guardian, which is a nonprofit organization with the mission of advancing genome-based newborn screening. And they have a goal of sequencing 100,000 babies in New York City. We were very delighted that Dr. Hugh was a guest on our podcast because he also shared his personal story of getting MPS II nominated to the recommended uniform screening panel, as we know as RUS. And in this podcast, he shared his personal story of his 2 children with MPS II and how, by documenting their experience led to a case study article, which really pushed forward the nomination package. So please definitely listen to his story and also share the podcast. The next slide. And please subscribe. Actually, we have one more, right, with the symposium. Did that get added?
Let me pull that up while you're talking
Okay.
about
So yes, so definitely do check out Dr. Hugh's podcast, and also last month's podcast was with Dr. David Bick, who shares his experience on Genome England, where their goal is to sequence I think over 100,000 babies in the UK. So you can see that these two podcasts were kind of positioned side by side to look at the US experience and the UK experience in the future. So I'm very happy to share with you that the inaugurated Harvey Levy Newborn Screening Symposium that's happening in December 8, 2002 from 8 a.m. to 4:30 p.m. Eastern time, because they're in Boston. And this is a free event. And you can see here, you can use your phone to scan this QR code, or you can just click on the link to here, which is the Google form that you can just sign up and you'll get the link to this symposium. And I'm also happy to share that MBS Strand, Dr. Brown will be part of this symposium. So we're very excited that MBS Strand is going to be represented at the symposium. So definitely please check this out. We're going to do a huge blast sharing this through our social media channel. But if you can also share this symposium through your networks, that'd be great. I think that's the last slide. And then the other one was just to follow us on social media. And then the next webinar, Amy, is the first Thursday of December. So we're going back to our original time slot. We only changed it this month because of the advisory board meeting. So just make sure everybody know it's going to be December 1st, 2022 at 2 p.m. Eastern time. Any
Great, and
questions?
we're excited to bring experts like Dr. Bedoyan to connect the newborn screening community with clinical researchers. We would love to have your feedback on the structure of these talks. I know we always seem to run out of time because there's so much to share on these hour-long talks. So we would love to, again, keep the conversations going and to get your feedback on these important conversations. And we can definitely leave open the Zoom. I know, Dr. Bedoyan, you've been answering a few questions in the chat. So if anybody wants to stay on and continue to talk, feel free to do that.
Thank you very
Assuming,
much.
Dr.
everybody.
Bedoyan, you don't have to run off and see patients. So
Yeah, I do have things to do, so they're welcome to send me an email, and I would be happy to respond to their questions in person.
great.
So you can share with them my email, and then I'll be happy to follow up any questions.
We're happy to do that. So thank you so much, Dr. Bedoyan. Thank you so much, Dr. Chan, and have a great rest of your day, everybody.
Thank you.
Thank you.